This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy, we go deep and lightly surface the big ideas. I want you to picture something for a second.
It's okay.
Picture the ancient European landscape.
Right, we're talking deep history here.
Exactly, deep history. Specifically, that really crucial window right between the 6th and 3rd millennium BC.
Ah, the Mesolithic and Neolithic transition.
Yeah, the transition. And if you're familiar with the period, you know this is a time of just immense environmental and cultural shift.
Massive shifts.
Right, but when it comes to the actual daily lives of the people who live there, you know, in these dense forests and river basins, our collective imagination almost always falls back on this very persistent, very specific image.
Oh, absolutely. The caveman stereotype.
Right. Even those of us who study history, we tend to picture these harsh survivalists.
Mostly hunting.
Mostly hunting. The cultural shorthand is basically this carnivorous hunter-gatherer just tearing into a roasted mammoth leg beside a fire.
A very protein-heavy image.
Very protein-heavy. It's this compelling picture of human resilience.
It is a pervasive image. And honestly, it's one that has dominated popular culture and frankly, early archaeological interpretations for over a century.
Wow, that long.
Yeah. We've historically visualized prehistoric Europe as this unforgiving tundra or, a dense boreal forest where survival was just intrinsically linked to hunting large megafauna.
Right, or mass harvesting fish.
Exactly. The whole gatherer, half of the hunter-gatherer equation, has for a very long time been relegated to this secondary, almost incidental role in our historical narratives.
Like it was just a side hustle.
Absolutely, yes.
And that bias is exactly what we are dismantling today. Welcome to a deep dive that completely rewrites the prehistoric menu.
I am very excited about this one.
Me too. We have this fascinating stack of research in front of us, and it's all anchored by a groundbreaking 2026 study published in the journal PLOS One.
Led by Laura Gonzalez-Carretero and her colleagues.
Right, from the University of York. And what they've done is conduct this exhaustive microscopic analysis of ancient pottery.
From 13 different archaeological sites.
13 sites. And these aren't just concentrated in one tiny area either. They span a massive geographical transect across northern and eastern Europe.
Which is such a brilliant approach.
Why is that?
Because it gives us a panoramic view of human adaptation. We're looking at communities that occupied vastly different ecological niches.
From like modern-day Russia to the Baltic Sea.
Exactly. From the riverine systems of Russia all the way to the coastal lagoon environments of the Baltic.
Right.
And what this study does so effectively is challenge the assumption that these communities were simply subsisting.
Right. And the mission of this deep dive is to explore how these early European hunter-gatherer fishers were, in fact, highly sophisticated culinary innovators.
Master chefs, really?
Yeah, they weren't just throwing chunks of meat on a fire or eating raw fish out of pure necessity. They were developing complex, regionally distinct, and incredibly plant-rich recipes.
They understood the chemical properties of their local flora.
And they used pottery to actively manipulate flavor, texture, and even toxicity.
Okay, let's unpack this. Because to understand how revolutionary this 2026 study is, we first have to understand why the scientific community had this massive, meat-heavy blind spot for so long.
What's fascinating here is that the blind spot wasn't a result of scientific negligence.
It wasn't.
No, not at all. It was an inherent limitation of the surviving evidence. In archaeology, we constantly battle against something called taphonomic bias.
Taphonomic bias. Explain that for the listener.
Taphonomy is essentially the study of how organisms decay and become fossilized or preserved.
Okay.
So for generations, the primary method for reconstructing ancient diets was zooarchaeology.
Which is looking at animal bones.
Right, the identification and quantification of animal remains.
Because animal remains, specifically bones, teeth, antlers, they're composed of calcium phosphate.
Exactly. And under the right soil conditions, particularly in non-acidic environments, that inorganic matrix can survive for thousands of years. So an early archaeologist would excavate a site, uncover these massive middens.
Ancient trash heaps.
Right, these ancient trash heaps full of deer bones and fish scales, and they'd logically conclude, well, these people ate an overwhelming amount of meat.
Precisely. The taphonomic bias creates a survivor bias.
Because the plants disappear.
Yes. Plant matter is primarily composed of cellulose and water. In the vast majority of soil conditions, a leafy green, a tuber, or, a handful of berries, they'll succumb to microbial degradation in a matter of weeks.
They simply vanish.
They vanish.
Yeah.
So the absence of physical plant remains was frequently misinterpreted as an absence of plants in the diet.
Wow.
But even as science progressed, we still struggled to find the plants, right? Let's talk about lipid residue analysis.
Yes, the game changer.
Because for the last few decades, that has been the gold standard for figuring out what ancient people cooked.
It has.
And on paper, it sounds like absolute magic. I mean, you find a broken piece of a 7,000-year-old clay pot, you grind up a tiny sample, and you extract the fats.
The lipid.
The lipids, right. The lipids that soaked into the porous clay during cooking.
It was a massive leap forward. The technique primarily relies on gas chromatography, mass spectrometry.
GCMS.
ECMS, exactly. To simplify a highly complex process, the researchers vaporize the extracted residue and pass it through a column.
Which separates the individual molecules based on their chemical properties.
Right. Then the mass spectrometer breaks those molecules apart and measures their mass-to-charge ratio. This gives us a highly specific chemical fingerprint.
A biomarker.
Biomarker.
And those biomarkers are incredibly good at identifying animal fats, aren't they?
Oh, phenomenally good.
Like, if a Mesolithic community was boiling down freshwater carp or cooking a deer stew, the marine oils and the adipose fats leave very strong, distinct chemical signatures.
Things like specific ratios of palmitic and stearic acids.
But here is the critical flaw in the GCMS methodology when it comes to the full dietary picture. Plant lipids degrade entirely differently than animal fats.
That is the crux of the issue. The chemical signature of a boiled root or a leafy green is incredibly faint compared to the heavy triglycerides of animal fat.
It just gets drowned out.
Exactly. The study notes that plant biomarkers do exist, things like very long-chain fatty acids or specific triterpenoids and phytosterols.
Like alpha-amarin and beta-amarin.
Right. But these compounds are highly susceptible to degradation over millennia. Furthermore, even when they do survive, they offer incredibly low taxonomic resolution.
Meaning that even if the mass spectrometer detects the presence of a plant lipid, it often can't tell you what kind of plant it was.
Exactly. It can't tell you if that lipid came from a wild onion, a berry, or a blade of grass. It just says plant.
Just plant. So if you are looking at a pot where someone cooked a rich, fatty salmon alongside a handful of delicate wild greens,
the animal fat completely overwhelms the data.
the science was effectively wearing meat-colored glasses.
That's a great way to put it. It was washing away the culinary complexity of these ancient meals.
Which brings us to the methodological breakthrough of the York team. Because to circumvent this chemical bias, they turn their attention to something that, ironically, modern cooks absolutely despise.
The food crust.
Food crust. We've all been there. You leave a stew on the stove for too long, the water boils off, and you are left with this charred, cemented black layer on the bottom of your pot.
The absolute worst to clean.
Today, we view that as a ruined pan that requires overnight soaking and a wire brush. But to the archaeologists in this study, that amorphous carbonized deposit is a holy grail.
It really is. The carbonization process is what saves the evidence.
How does that work?
When organic material is burned in a low oxygen environment, like the bottom of a simmering clay pot, it carbonizes. This stops the normal bacterial decay process in its tracks.
It just freezes it.
The heat essentially bakes the microscopic cellular structure of the food into a hardened matrix of carbon. It becomes a localized microscopic time capsule.
But the researchers didn't just run these crusts through the GCMS machine to look for chemical fats, right?
No, they deployed a dual methodology approach.
They integrated the chemical analysis with high-resolution digital and scanning electron microscopy.
CM.
CM. They took 85 individual pottery sherds featuring these thick, burnt crusts from those 13 sites, and they physically looked at them.
And we should clarify just how powerful a scanning electron microscope is for those who might not work with them.
Yeah, paint a picture of this machine.
Unlike a traditional optical microscope that uses light to magnify an image, and SIM uses a focused beam of electrons.
Okay.
These electrons interact with the atoms in the sample, producing varied signals that contain information about the surface topography and composition.
And the magnification is insane, right?
It allows researchers to achieve magnifications of up to 500,000 times,
Wow.
creating incredibly crisp, three-dimensional images of structures at the nanoscale.
So they're zooming in so closely that they can observe the individual cell walls of a fragmented, 7,000-year-old seed embedded in a matrix of charred fish fat.
Yes. And when they did this, the findings were staggering.
What did they see?
Embedded within these crusts, they found the preserved fossilized tissues of wild grasses, legumes, fruits, roots, tubers, leaves, and stems.
The prehistoric side salad was finally visible.
It was finally visible.
But identifying a charred cellular structure under a microscope is only half the battle, isn't it? I mean, how do you definitively prove that an anomalous clump of cells is, say, a specific type of wild legume that has been boiled and charred?
I'm so glad you brought this up because this is where the study gets incredibly rigorous. To validate what they were seeing under the electron beam, the researchers had to engage in extensive experimental archaeology.
They literally had to build a prehistoric kitchen.
They did.
But I have to ask, when we talk about experimental archaeology, how do we know their replica pots and modern wood fires accurately match a cooking environment from 5000 BC? Isn't there a significant margin of error there when trying to recreate ancient chemistry?
It is a vital question, and it's where the meticulousness of the experimental design matters. They didn't just throw modern clay into a bonfire.
Right.
They crafted replica vessels, utilizing the exact compositional analyses of the ancient ceramics from the region.
So matching the temper, the clay types, the porosity.
Exactly. They then built open wood fires and allowed them to burn down to stable, active embers.
Which mimics the controlled cooking fires we see in the archaeological record rather than just raging bonfires.
Precisely. And they controlled the temperature variables meticulously.
The study notes they use thermocouples to monitor the heat.
Yes, maintaining a steady internal temperature between 120 and 160 degrees Celsius.
So they were essentially slow boiling these ingredients.
They took common freshwater fish from the region, like carp, and stewed them together with wild ingredients they foraged themselves.
Things like guelder roseberries and goosefoot stems.
Yes. They then intentionally charred these mixtures, moving the pots closer to the heat source to induce the formation of the food crust.
So this wasn't just performance art.
No, this was about creating a definitive modern reference library. By placing these experimentally charred crusts under the SEM, they could document exactly how the cellular structures of a guelder roseberry fracture, deform, and preserve when subjected to the specific thermal dynamics of boiling and burning in a clay matrix.
They basically created a Rosetta Stone of prehistoric burnt food.
That's exactly what they did.
And when they cross-referenced the cellular deformation of their modern experimental stews with the 7,000-year-old crusts, they had a morphological match.
They didn't just find stray plants, they found evidence of deliberate, complex cooking techniques.
It definitively proves that these ancient communities were selectively gathering specific plant parts and combining them with specific animal proteins.
It demonstrates a level of culinary intention and environmental mastery that just forces us to completely reevaluate the cognitive and cultural complexity of Mesolithic and Neolithic peoples.
Which perfectly transitions us into the menu itself, because the study doesn't just reveal a monolithic ancient diet.
Far from it.
It takes us on a culinary tour of Northern and Eastern Europe, showcasing how deeply these cuisines were tied to the specific ecologies of their regions.
It's fascinating to see the regional variations.
Let's head to our first destination, the riverine ecosystems of the upper and middle Don River Basin.
Located in modern-day Russia.
Right. We're looking at sites like Cherkasskaya 3 and 5. What did the microscopic detectives uncover in the pots from the Don River?
At the Don River sites, the SM analysis revealed a striking reliance on wild grasses.
Specifically members of the Poaceae family, right?
Yes, and wild legumes, which were consistently cooked alongside freshwater fish.
Mostly sprinnids.
Right, which belonged to the carp family.
Now, when we talk about wild grasses, we aren't talking about early agriculture, right?
Oh, no.
These are purely foraged, undomesticated ancestors of the grains we recognize today.
Correct. These are entirely wild stands of grasses. But what makes the SEM findings so remarkable is the sheer anatomical detail preserved in the crusts.
The researchers were able to identify the pericarp cell tissues, right?
Yes, the outer layers of the grain. They specifically noted the transverse and longitudinal cells that constitute the bran.
Wow.
Even more critically, they identified single-celled aileron layers.
Okay, let's break that down for you listening, because the aileron layer is a fantastic piece of botanical forensics. The aileron layer is a tissue found in the seeds of grasses, situated right between the starchy endosperm and the bran. It's packed with proteins and nutrients. But why is the fact that it's single-celled matter so much to the researchers?
It is a vital taxonomic discriminator. Different families of grasses have differently structured aileron layers.
Oh, I see.
For example, wild barley, wild oats, and certain brome grasses typically have multi-layered aileron structures.
So by finding a single-celled layer,
By definitively identifying a single-celled layer in the charred remains, the researchers can immediately rule out those species. It rapidly narrows down the botanical candidates, pointing strongly toward other specific wild grain varieties endemic to the Don River Basin at that time.
that level of specificity from a millennia-old burnt speck is just astounding.
It really is.
And alongside these specific grasses, they found legumes at the Cherkasskaya 3 site. The microscopes revealed the seed coats of small, wild legumes featuring very distinct palisade and hourglass cells.
And based on the morphology of those cells, the researchers concluded these were likely wild clovers.
Wild clovers, which highlights a massive investment of labor.
Absolutely.
Because processing wild grasses and small legume seeds like clover is an incredibly tedious task. It requires systematic gathering, threshing to remove the chaff, winnowing, and ultimately prolonged cooking.
Right, because humans cannot easily digest raw, unprocessed grass seeds.
We just don't have the stomachs for it.
No. Cooking them in a ceramic vessel with fish creates a gelatinized, nutrient-dense pottage, making those complex carbohydrates biologically available.
So in the Don River Basin, we have this hearty, labor-intensive, slow-cooked stew of carp, wild grains, and clover. Sounds almost modern, like a porridge.
Very much so.
Now, let's travel northwest to our second culinary region, the Upper Volga and Dmardvina area.
This encompasses sites like Zamoster II and Sirteya II.
Here's where it gets really interesting, because the ingredients shift dramatically from wholesome grains to something much more challenging.
Indeed. In the Volga and Dmardvina regions, the dominant plant signature dominating the food crusts belonged to Viburnum opulus.
Commonly known as the Gelder roseberry.
The Gelder roseberry, yes.
I spent some time researching the Gelder roseberry before we recorded, and it is a fascinating, if somewhat alarming, ingredient.
It's not your typical berry.
No. If you forage these berries and eat them raw, they are mildly toxic. They contain compounds that act as severe gastric irritants. Furthermore, they are intensely bitter and highly astringent.
The researchers even noted in their experimental archaeology phase that boiling these berries released a remarkably pungent, unpleasant odor.
Some ethnographic accounts compare the smell to sweaty feet or wet wool. So, why on earth would a community systematically choose to harvest and cook a toxic, foul-smelling bitter berry?
That is the pivotal question of human behavioral ecology. If the primary driver of foraging is simply caloric intake, there are far less demanding sources of energy in a Holocene forest.
Right.
We know from the broader archaeobotanical record, the raw seeds and pollen preserved in the soil strata at these sites, that a wide variety of flora existed.
There were other options.
Plenty of options. Yet the crusts inside the cooking pots show a deliberate, repeated, and highly specific pairing of these bitter viburnum berries with freshwater fish.
It wasn't an act of desperation, it was a curated recipe.
Exactly. And the botanical complexity doesn't stop with the berries. In these same Volga region pots, the same analysis revealed a heavy reliance on plants from the Amaranthaceae family.
Which includes species like goosefoot and various salt bushes.
Yes.
Goosefoot is essentially the Mesolithic equivalent of spinach or Swiss chard, but there has always been a debate in the archaeological community about these types of plants, right?
The weed versus food debate.
Because they are ruderal weeds, they thrive in disturbed soils, exactly like the nitrogen-rich soil you'd find around a human settlement or a campfire.
Yes. Because these plants naturally aggregate around human camps, finding their seeds in the soil doesn't prove they were eaten. They could have simply blown into the fire.
Just accidental contamination.
Right. But the SEM analysis of the food crust definitively resolved this. The researchers didn't just find stray seeds. They found the remains of entire inflorescences.
The complete flowering structures of the plant.
They found preserved calyces, leaves, and stem epidermis tightly embedded in the fish fat matrix.
They were actively harvesting the entire plant and throwing the greens right into the stew.
And the microscopic data even tells us when they were doing it.
This part blew my mind.
By analyzing the developmental stage of the preserved plant parts, the researchers found green, underdeveloped calyces that only appear in May and June.
Mixed in the very same vesselants with fully ripe calyces and embedded seeds that develop in late July and August.
Which is a profound piece of evidence. It tells us that this wasn't a singular panicked autumn harvest to stave off winter starvation.
Right.
This was a continuous, seasonal culinary practice. These communities intimately understood the life cycle of the Chenopodium plants. They returned to these specific greens week after week, month after month, consistently utilizing them to flavor and bulk out their fish stews throughout the entire spring and summer.
That level of intimate environmental literacy is incredible.
Truly.
We've seen the grain stews of the Don and the bitterberry and seasonal green stews of the Volga. Now let's move to our final destination, which offers perhaps the most dramatic shift in diet, the Baltic region.
We are looking at sites dotting the coasts of modern-day Denmark, Poland, and Lithuania.
Associated largely with the Erdebol culture.
The Baltic Sea presents a massive geographical and ecological divide, and we see that reflected immediately in the GCMS lipid residue data.
How so?
In the eastern Baltic sites, such as those in Lithuania, the chemical biomarkers are overwhelmingly dominated by freshwater fish fats. However, as we move to the western Baltic, to the Danish sites, the chemical signatures pivot sharply. We see a massive influx of marine biomarkers.
Sea fish, marine mammals like seals.
Exactly. Coupled with the adipose fats of ruminant animals like red deer or roe deer.
The classic surf and turf.
Yes. But the plant component is what truly stands out here, particularly at the site of Silholm II in Denmark. The microscopic analysis of the crusts from this coastal settlement yielded something incredibly rare in the Mesolithic archaeological record.
The SEMIM imagery at Silholm II revealed distinct, anomalous vascular tissues paired with specific parenchyma cells.
Yes.
Let's explain parenchyma for the listener because it's a critical piece of the puzzle. Parenchyma is essentially the versatile foundational ground tissue in plants. It's the soft, spongy cellular material that makes up the fleshy interior of an apple or the bulk of a root vegetable. But what makes the tissues at Silholm II anomalous?
When analyzing the cross-sections of these charred remains, the researchers observed a highly specific pattern of secondary growth. They documented concentric rings of vascular tissue.
The xylem and phloem that transport water and nutrients.
Right, and these rings were separated by broad, distinct bands of storage parenchyma. In botanical terms, this specific cellular architecture is the definitive morphological signature of underground storage organs.
We're talking about roots and tubers.
They were cooking prehistoric root vegetables. Based on the cell measurements and the paleoenvironmental data of the Danish coast, the researchers hypothesized they were likely processing things like sea beet.
Which is the wild coastal ancestor of modern beetroot and Swiss chard.
Exactly. They also identified structures consistent with sea club rush tubers.
Characterized by solid parenchyma tissue with scattered vascular bundles.
Yes. And we know from previous excavations in the region that they were also actively foraging wild garlic bulbs.
So we can begin to reconstruct the sensory reality of a meal at Silholm II. You have marine fish, perhaps the rich fat of a seal or a deer, combined with wild garlic and the dense carbohydrate-rich flesh of sea beet tubers, all slowly simmering in a ceramic vessel.
It demonstrates an incredibly sophisticated, multi-stage manipulation of the local coastal biosphere.
And there's a fascinating twist at Silholm II regarding the lipid analysis.
And the dairy.
Yes. Alongside the marine and ruminant fats, the GCMS data picked up the distinct biomarkers of dairy products. Now, we're talking about Mesolithic hunter-gatherers. They did not have domesticated cattle or sheep to milk.
They did not. But the chronological context is vital here. By the 4th millennium BC in the western Baltic, these indigenous foraging communities of the Erdebol culture were living in increasingly close proximity to early Neolithic farming groups.
Who were migrating into the fringes of Europe, bringing domesticated livestock with them.
Right, so the presence of dairy fats baked into the crusts of hunter-gatherer pots is chemical proof of interaction.
They were trading with the farmers, acquiring exotic ingredients like milk or butter and integrating them into their traditional coastal root and fish stews.
It's the dawn of prehistoric fusion cuisine.
It's amazing.
It speaks to the fluidity and adaptability of these cultures. But I want to pivot back to the eastern side of the Baltic, to a site called Nivkh on the Pomeranian coast of modern-day Poland.
Because at Nivkh, we see a fascinating anomaly regarding our old friend, the bitter viburnum berry.
We do. When archaeobotanists analyze the general soil samples excavated from the settlement area at Nivkh, they find ample evidence of starchy local plants, like the roots of the flowering rush.
But what they do not find in the general environmental samples is any trace of viburnum berries. They were not growing abundantly around the camp.
Yet, when the researchers analyzed the food crusts, specifically from the interior of the cooking pots at Nivkh, The microscopic remains of viburnum berries were not only present, they were frequently and explicitly mixed with high concentrations of freshwater fish biomarkers.
So what does this all mean? If a highly specific toxic bitter berry is completely absent from your immediate environment, but it consistently shows up inside your cooking pots perfectly paired with fish fat, you are going out of your way to procure it.
If we connect this to the bigger picture, it proves that this was not opportunistic foraging. The people of Nivkh were either undertaking targeted logistical expeditions to specific ecological zones to harvest these berries, or they were acquiring them through trade networks.
They expended significant caloric energy to obtain an ingredient that was, on its own, toxic and unpalatable.
They did this because their specific culinary tradition demanded it.
Which brings us to the profound realization at the heart of this research. We've established the what, the grains, the tubers, the toxic berries, but we have to address the why.
The motivation.
Why go to the immense trouble of crafting highly specific ceramic vessels, controlling fire temperatures to a precise degree, and laboriously processing toxic berries and wild grasses? It fundamentally shifts our understanding of why pottery was adopted in the first place.
For decades, a dominant theory in prehistoric archaeology was that early European hunter-gatherers adopted pottery for strictly utilitarian industrial purposes. The prevailing hypothesis was that these communities needed large, heat-resistant vessels to boil massive quantities of fish in order to render down the fish oil.
And this oil was hypothesized to be used as fuel for lamps, as waterproofing sealants, or perhaps for preserving surplus food.
It paints a very bleak, hyperfunctional picture of early human life, just boiling vats of fish fat to keep the lamps lit.
But the microscopic evidence from this PLOS One study directly contradicts that utilitarian reductionism.
The researchers explicitly state that we can now confidently dismiss the possibility that European forager pottery had a predominantly non-culinary role.
These ceramic pots were not just industrial rendering vats. They were essential, highly evolved culinary tools.
The invention and widespread adoption of pottery allowed early humans to act as chemists, fundamentally altering the taste, texture, and toxicity of their environment.
Let's look at the chemistry of that bitter viburnum berry stew as the perfect example. You have a berry that will cause severe gastric distress if eaten raw. But when you introduce it to a ceramic pot filled with water and fatty carp, a transformation occurs.
Boiling the berries in water is the first step. The thermal energy helps to break down the rigid cellular walls and leach out some of the water-soluble toxic compounds.
But the introduction of the fish fat is the true culinary genius.
Absolutely. Lipids are incredibly effective at enveloping bitter and astringent compounds. The fat physically coats the molecules, preventing them from binding to the bitter receptors on the human tongue.
What was once a foul-tasting, toxic berry is neutralized.
It's transformed into a rich, calorie-dense, and potentially complex sweet or savory component of a meal.
They were actively using fish fat as a chemical buffer to unlock a previously inaccessible food source. And one of my absolute favorite parts of this study is that the researchers found a living ethnographic parallel for this exact chemical process.
Yes, to understand the ancient Volga River communities, we can look at the modern Nivkh people.
The ethnographic data provides a stunning bridge across millennia. The Nivkh are an indigenous ethnic group inhabiting the Russian Far East, primarily near the estuary of the Amur River and on Sakhalin Island.
Culturally and historically, they are heavily reliant on fishing and foraging.
And for generations, the Nivkh have prepared a highly specific traditional dish known as mos.
And mos is essentially a fish and berry jelly.
The traditional preparation involves taking dried fish and pulverizing it. They then boil fish skins, which are rich in collagen, releasing a natural gelatin.
They mix this vigorously with the pulverized fish, various local berries, and sometimes tubers or roots.
As the mixture cools, the fat and collagen cause it to set into a dense, nutrient-rich, sweet and savory jelly.
It is a dish that utilizes the exact same principles of lipid buffering and carbohydrate protein synthesis that we see in the 7,000-year-old pots from the Volga and Dnieper regions.
The ancient communities boiling their carp and their bitter gelder roseberries were likely creating a Mesolithic precursor to moses.
It completely reframes how we view these people. A fish and berry jelly might not be on the menu at a modern bistro, but you can instantly recognize the culinary intent.
This isn't desperate, shivering survival. This is the deliberate crafting of a cherished cultural dish.
It represents the birth of deep culinary tradition, and it requires profound generational knowledge transfer.
Right, because you can't just guess this stuff.
Exactly. A community has to pass down the knowledge of exactly which plants are toxic, raw, but nutritious when boiled with specific animal fats. They must teach the next generation that the green calices of the goosefoot plant harvested in May yield a different flavor profile and nutritional value than the seeded calices harvested in August.
They have to teach the visual cues required to identify the anomalous vascular tissues of a sea beet tuber buried beneath the coastal soil.
This level of environmental mastery is staggering, and it was entirely invisible to us until we looked through the right microscope.
It makes you realize that they weren't just passively reacting to their environment, they were actively orchestrating it. They had a desire for complex flavor profiles, they sought out specific textures.
They were cooking for comfort, for identity, and for tradition.
With the same intentionality that we approach a complex family recipe today.
This raises an important question, however, about the fragility of our historical narratives.
How so?
The findings of this single study, by simply changing the methodological lens from chemical lipid extraction to scanning electron microscopy, have completely upended a century of assumptions regarding Mesolithic and Neolithic diets.
It is the ultimate lesson in the dangers of taphonomic bias. Because the bones survived, we assume they were exclusively hunters. Because the heavy animal lipids survived the chemical extraction process, we assume they used pots only for meat and fat.
The plants, the delicate tubers, the seasonal herbs, the very ingredients that brought color, flavor, medicine, and cultural identity to their lives, decayed into dust.
And for decades, the scientific community misinterpreted that absence of evidence as evidence of abscess.
It serves as a potent reminder that our understanding of the past is only ever as complete as our methodologies allow. We must constantly interrogate the trash of the past, the burnt mistakes, the amorphous crusts, with increasingly sophisticated tools.
The most profound truths about human history are frequently hidden in the materials we previously overlooked or discarded.
Absolutely.
Which brings us to a fascinating final thought for you to ponder. We've spent this entire deep dive dismantling the myth of the carnivorous, meat-exclusive caveman. We've proven that these ancient European hunter-gatherers were actually master botanists, heavily reliant on complex carbohydrates, wild grains, and diverse plant matter.
That is the clear conclusion of the data.
Yet, if you walk into any supermarket or open any fitness magazine today, you are bombarded by the modern paleo diet industry.
Oh, yes.
It is a multi-billion-dollar wellness empire built almost entirely on the premise that our ancestors thrived on a low-carbohydrate, hyper-carnivorous diet of pure meat and fat.
It is a striking disconnect. The modern dietary trend has commercialized a version of prehistory that is rooted entirely in the very taphonomic biases and outdated zooarchaeology that modern microscopy is actively debunking.
Exactly. If the burnt microscopic crust scraped off the bottom of a broken 7,000-year-old clay pot proves that ancient humans were sophisticated processors of wild grains, tubers, and complex carbohydrates. What does that mean for a modern diet industry that rejects those exact foods in the name of ancestral health?
It makes you wonder how much of our modern wellness culture is based on a phantom version of history that never actually existed.
It is a compelling contradiction, and it highlights how eagerly we project our modern desires onto the distant past.
We truly do.
Thank you for joining us on this culinary journey through deep time. We hope you walk away with a profound new respect for the ancient chefs who engineered the foundations of cuisine and a healthy skepticism of anyone trying to sell you a prehistoric menu. Keep questioning the narrative, stay curious, and we will see you on the next deep dive.
Heliox is produced by Michelle Brucker and Scott Bleakley. It features reviews of emerging research and ideas from leading thinkers, curated under their creative direction with AI assistance for voice, imagery, and composition. Systemic voices and illustrative images of people are representative tools, not depictions of specific individuals. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other episodes, responding to the content, and checking out our related articles at helioxpodcast.substack.com.